All the ventricles located here. We have the two lateral ventricles, these big C-shaped items on the side. And I've been trying to tell you guys, they're large. They're C-shaped, large ventricles that connect through very small foramen to the third ventricle, which is right in the middle. And the third ventricle through the cerebral aqueduct. Looks like a little seahorse down to the fourth ventricle with its tiny, ineffective arms. Out of the median aperture, the two lateral apertures, and then to the central canal in the spinal cord. And you can see some cool stuff if you, let's say we hide or fade this, and then zoom in again. You can really start to see the choroid plexuses. No, you can't. They're not on this one. That's great. Because I introduced that, I want to just bring it up for you. Let's say we have the choroid plexus in the left ventricle and the right ventricle. And then let's close this up. Oh, of course it wouldn't work. Okay, never mind. They line the interior surface all the way along the top, down through the bottom, and then all the way through the third and fourth ventricles. So it helps to position the choroid plexuses within the ventricles to help with the understanding, I think, if it would resolve for me. So we'll leave that for now. It's not terribly user-friendly as it stands. As we continue with our discussion. So after the brainstem, and what were the three subsections of the brainstem? From the spinal cord, we move right to the, I heard it over here, medulla, yep, then pons, then midbrain. Fantastic. You guys are reviewing well. That's good. For those of you in lab yesterday that got the, I won't say anything, never mind, but you've been prompted to go back and review. After the midbrain, we have what we call the diencephalon. The diencephalon is composed of three main parts itself. By far, the definitive, most defining factor is the thalamus, which is the olive-shaped prominence right in the middle, underneath this light white-colored C-shaped thing that we'll talk about today as well. Thalamus, easy to see, especially in the sagittal sections of the brains that you will have in lab this week. The hypothalamus and the epithalamus, not as easy to see. And I'll show you where you might be able to find them. In general, they're all sort of in this area. The hypothalamus is not so much a prominence as it's a surface. It's the surface of the third ventricle, just below the thalamus. And we'll get into that as we go through the sections on each of these. The three main parts to the diencephalon, largest, most identifiable is the thalamus. Hypothalamus, which we've registered or talked about before, and then the epithalamus. Also a projection of the diencephalon is the pituitary gland. You can see the pituitary gland hanging off here, connected to the infundibulum. On some of the brains, the whole sections with the meninges intact, you can see this still in lab, which is really interesting. But the other ones, it seems to have been removed or worn off or cut off. And largely this is referred to as the master gland, the pituitary gland. It controls metabolism and many hormonal secretions, growth, pregnancy, kidney function, etc. Major endocrine gland, the pituitary gland. The role of the diencephalon is largely to filter out those senses or to permit sensory information that needs to go to your conscious awareness. So it's a relay for afferent or sensory information entering the cerebrum. And when we think of cerebrum, we think of being knowledgeable about, knowing, being conscious of. It's within your awareness. So if senses are allowed to pass through to the cerebrum, you're aware of those senses. You're aware of a hot dish. You're aware of stepping on a nail. You're aware of something moving off to the side. So the thalamus being the major part, the largest part of the diencephalon, is shown here. It's within the brain, surrounded by the cerebrum, and it contains the third ventricle. Well, the diencephalon as a whole contains the third ventricle. Largely, the thalamus is the majority of that. It's around the third ventricle. It might be 80% of the diencephalon. It's large. And what's not important necessarily on this slide are all the colors and the convolutions of the cerebrum yet. We'll get into those. They're not immediately important right now, although just note that there are different colors. We'll talk about what they mean in a second. And note the location of the thalamus here, right in the middle. There are two lobes, a lot like the brain overall, the cerebrum. There are two parts that are connected by what's called the interthalamic adhesion, just the bridge between the two lobes. And if we zoom in, we see a bit more detail. Now watch the colors here, and then see the colors on the thalamus. This simply implies that there are connections between these regions of the thalamus and the larger regions of the cerebrum. So signals from this light blue area here on the ventral posterior section go to the corresponding color of the cerebrum, which happens to be the postcentral gyrus, and we'll talk about what that means in a second. There are a lot of different sections, and really you don't need to know all the sections, but I do want to draw your attention to two of the smaller ones that are actually right out of the way. There's two nuclei tucked below and posterior on each side of the thalamus, the geniculate nuclei. And the lateral geniculate nucleus is involved in receiving visual information and relaying that to the brain. Visual information enters here. The lateral geniculate nucleus, or nuclei, there's two of them, relays auditory information. So any sounds that you hear are brought to your conscious awareness through those structures. And there are other connections, but they're pretty involved and detailed, and we're not going to explore them in too much depth. I will say, because we bring up the limbic system later, the emotional system, or the system that's typically related to emotions or thought to be involved in forming emotions, does connect to the ventral or the underside surface. And we can't see where exactly, but there are connections between the emotional brain, the emotional system, and the thalamus that brings those emotions to the higher order awareness. So a couple key structures, and notice the overlapping colors, that there are connections between this and the cerebrum at large. Second part of the diencephalon is the hypothalamus. The hypothalamus is not a discrete structure. It contains 12 nuclei. There's many different little pinpoints of activity that can be defined if you look closely enough. But more appropriately, these line the walls of the third ventricle. So you can see as we zoom in, the thalamus is obvious on the larger section in the top left, that olive shape, and then the inset figure zooms in just inferior and anterior to that region. You can even see the thalamus, the bulge here. There's the interthalamic adhesion. And it's the wall that contains the nuclei of the hypothalamus. This is important. The hypothalamus largely regulates homeostasis, all the things that we've talked about, heart rate, breathing rate, some role in metabolism as well, temperature, blood flow to different regions. What makes this special is that lining the surface, it's in contact with the cerebrospinal fluid. Or there are parts of it that are directly bathed in the cerebrospinal fluid. That is, they're not protected by the blood-brain barrier. So you have free observation of pH, glucose, ions, temperature from some of the structures in the hypothalamus. And we'll talk about what they're called specifically coming up. But they allow direct sensory information to translate into a downstream effect. For instance, one of the areas that I research related to dehydration involves shutting off blood flow to the skin. If you get hot, you want blood to go to the skin so that you can sweat and evaporate that heat. You can dissipate that heat. You saw that in the last section. But if there's ever a situation where you don't have enough fluid, and you're dehydrated, you can't send blood to the muscle and to the skin. You have to choose. And you choose largely thanks to the hypothalamus. When you get dehydrated, your blood gets salty. It concentrates. And so the cerebrospinal fluid gets salty and concentrates. And the nuclei in the hypothalamus sense that it's getting salty, and it turns off blood flow to the skin. That's why when people exhibit heat stroke or heat exhaustion, in some cases, they've stopped sweating because of this mechanism. They're called circumventricular organs, but there's a slide devoted to those coming up, CVOs. Doesn't really matter. There are 4 regions that encompass the 12 nuclei, but maybe I'll just decide right now I'm not going to ask you detailed questions about the nuclei. So just know that there are 4 regions: the mammillary, tuberal, supraoptic, and preoptic, but we don't need to go into depth about what each of those do. Largely, it simply describes where the nuclei are. It's a good exercise. Supraoptic. Supraoptic. Well, a supraoptic nucleus is above the optic chiasm. You'll see this in lab. Those of you that were there on Monday have seen it. The optic chiasm is the X, the crossing of the optic nerves, and the supraoptic nucleus is right above that. Preoptic nucleus is in front of that one. Anyways, their functions are not necessarily important, but those that we've discussed. The epithalamus is even harder to discern. It's even harder to discern. It's not so much that there is a structure. There is one defining structure that tells us what area we would be able to find the epithalamus, and that's the pineal gland. Pineal gland is located here, tucked away, just behind the thalamus. And that's the gland that releases melatonin. Releases melatonin to help turn off the reticular ascending system, to help turn off the senses that allow you to go to sleep. You'll find it sandwiched between the cerebrum and the cerebellum, right at the back of the midbrain. And if you remember last week, we talked about the superior and the inferior colliculi. There's 4 little bulges that help you respond to loud crashes or visual flashes before you're even aware of them. It's right in the center of those, just on the superior border. It's very obvious. It also receives sensory information from the retinas. And this is why blue light from your phones makes it harder to go to sleep. Blue light depresses melatonin release from the pineal gland. Which is why you have apps like Flux, or built into iOS now, the blue light dims as it's later in the day to help reduce the effect on melatonin and keep you sleepy. Also on this slide are the habenular nuclei, which is simply an input from the olfactory bulbs, or the sense of smell. And it's a connection point to the cerebrum, to memories in the brain, and is part of the reason why smells register such strong emotions and memories. There's another input for the olfactory senses to the cerebrum, and that's through the limbic system. And we'll talk about that coming up. That's why you feel comforted when you smell apple crisp baking in the oven, like Grandma used to make, or the smell of soap sometimes is really, it's like a strong signature. It reminds you of years gone past, who knows. Or your grandma might just have an old person smell. The specific locations in the hypothalamus that are bathed in cerebrospinal fluid, like I alluded to before and is written out here specifically, are the circumventricular organs, or CVOs. It's this characteristic of not being blocked off by the blood-brain barrier that allows them to sense chemical changes in the blood through the cerebrospinal fluid. Now, the main ones that I talked about are those that are in the hypothalamus, because that's what I'm interested in. But the pituitary gland and the pineal gland are also considered CVOs also. They're outside of the blood-brain barrier. I'm going to wait a bit. I wanted to show you the hierarchy and the layers, but maybe at the end, when we talk about the limbic system, because that's involved too, I'll just bring that app back over and we can take a look at all the layers. It's a little bit harder to manipulate than on my iPad right now. So any questions about that, just quickly before we move on? Diencephalon is a quick stopover. There's not a whole lot of time spent discussing it. But a few key structures and a few key responses that we want to know about in the diencephalon. No? Okay. Oh, I'm sorry, yeah.
So
my eyeliner is up.
There's cerebrospinal fluid that the CVOs are able to detect changes in the body?
Yes. Yes, so they are bathed in cerebrospinal fluid directly. They're on the walls of the ventricles. And that will be more clear when I get the model up and running as well. I almost need to buy like an Apple TV and just bring that in. Okay, let's talk about the cerebrum. The seat of intelligence, of consciousness, of reasoning, of logic, of reflection, of planning, of daydreaming, of anxiety. All these things that we can infer that are not the present situation that other simpler organisms cannot imagine. Perhaps a blessing and a curse. Cerebrum, the seat of intelligence. The cortex on the outside, and there are some nuclei internal or deep within the cerebrum, are mostly composed of gray matter, which we identified as the processing areas, the processing neurons. Billions of neurons in the gray matter of the cerebrum with a very distinctive shape. There's lots of ripples and folds and crevices. These are sulci and gyri, also called sulci and gyri. And then fissures, we'll show you those on the next slide. These are defining features that make the brain the shape and the orientation that we think of when we think brain. Gray matter on the outside, which tends to develop faster than the white matter internal tracts during development, and that's why the folds occur, the undulations and folds, because there's so much gray matter to accommodate that it forces the brain to develop in folds and undulations. The white matter tracts are inferior, under the cortex layer. These are myelinated axons. They're protected axons that allow quick and effective transmission. So these are the pathways, transmission pathways within the brain. And there are connections on the same side of the brain, between sides of the brain, between the brain and then the lower order structures and the rest of the body. One of the main defining features that you'll see in lab is the corpus callosum, italicized here. That's that big white C-shaped thing that we brought up earlier in the class. It's the main tract between both hemispheres of the brain. Easily identifiable in lab. One of the defining structures that helps you orientate yourself to where you are in the brain. So let's look at fissures, gyri, and sulci. Fissures are deep grooves. The first, most obvious fissure is the longitudinal fissure, straight down the middle of the brain, that separates it into two halves, into two hemispheres. This is deep, it goes all the way down. You can open up both hemispheres without ripping the brain and see the superior surface of the corpus callosum. The sulci and gyri are all of these noodly undulations. The rises, the bulges, the hills are the gyri, and then the small grooves that are often defined or bordered by small blood vessels are the sulci. You can see them in the section here where we have one sulcus. It's not very deep, but it does increase the surface area for the cortex versus the fissure, which goes all the way through, separating into left and right hemispheres. Also notice the gray matter versus the white matter. Only the surface is the cortex or gray matter. And we're also getting some indication here about the functional divisions of the brain into lobes. So we thought hemispheres before. You've seen even more detail in the color shaded sections when we talked about the thalamus. Here we start to see lobes. There are larger sulci, or more defined sulci, that separate the brain into lobes. So we have the anterior portion, the frontal lobe, parietal lobe, occipital lobe, which are named for the bones covering them. Not for their functions, not for any other reason. They're named for the bones covering them. Each hemisphere has the same number of lobes and distribution of lobes. I kind of think of each hemisphere as a mitt. It looks like a mitt, like you'd wear in the winter. With the thumb being the temporal lobe on the far right, outside, or the lateral aspect. And then the meat of the hemisphere, the fingers and the palm, are the frontal, parietal, and occipital lobes as you go from anterior to posterior. Temporal lobe on the lateral aspect, frontal, parietal, and occipital. You won't be able to discern these in lab. Sheep brains are a lot smaller and harder to differentiate the sections of the lobes than human brains. There's a really nice video at the start of lab that keep an eye out for these lobes when you watch that video, without spoiling it too much. Also note the divisions between the frontal and parietal lobes are the central sulcus here. That's the only one that's not really obvious. The lateral sulcus, well, fairly obvious. It's lateral. It divides two lobes that are lateral, the temporal lobe being the most lateral. And then if I told you what the parieto-occipital sulcus was, you could reason out where in the brain that structure would be. Parieto-occipital sulcus. Parieto-occipital. So parietal on one side, occipital on the other. It's probably the division between those two lobes, and it's a sulcus, so it's a small groove here defining those two lobes at the back of the, or the posterior aspect of the brain. We also see the insula, but we don't really care about that. We think it's involved in emotion and consciousness, but we don't really know. It's between the thumb of the mitt and the hand. When we dive in, when we cut into the brain, here along a mid-sagittal section, and we see a real brain, we don't just have stylized gray and white matter. We can actually start to see some of the tracts some of the myelinated axons. So anterior on the right, posterior on the left. Look for defining factors. Right away, you can see sulci and gyri, the undulations. You can see gray matter, white matter. You can see corpus callosum. This structure here always looks like that. Easy to orientate yourself. And from there, you can start to tease out thalamus, hypothalamus, pituitary gland if it's there, which it doesn't look like it is. So the white matter tracts. What I want to get across in this slide are the connections between areas in the cortex. There's three kinds of connections. Three main kinds of connections. One we're going to call association tracts, which convey signals between areas on the same side of the brain, within the same hemisphere. These generally move anterior to posterior within the same hemisphere. They don't cross over. Crossing over is a commissural tract. The corpus callosum is the primary example of that. It is where signals are sent from one hemisphere to the other, right to left, left to right. It connects both hemispheres of the brain. Then projection tracts, much more vertical in nature, indicating they translate information from the cortex to the brainstem to the rest of the body, the lower regions of the brain, or lower order brain areas. And in general, this division holds up. Gray matter, superficial, white matter, deep. Except when it doesn't hold up, as is the case with the basal nuclei. The basal nuclei, also called the basal ganglia, that's the old-fashioned term for it. Basal nuclei is the more current term. And you can see the basal nuclei here. This blue area is the basal nucleus on one side of the brain. The purple area is the ventricle, the lateral ventricle. So it's contained almost by the inferior border of the lateral ventricle. And there are two main shapes or structures. You can divide this into two main parts. There's this lens-shaped part in the middle, and the border, which is connected to the lens, that looks like a tail. And we call them the lentiform nucleus, because that means lens, that big bulge in the middle, and the caudate nucleus, which means tail, because it looks like a tail. Two main regions, and then the further divisions of the basal nuclei I don't think are too important, but you can see them over on the right-hand side as we look through this frontal plane. You can see the lentiform section here, and there are multiple different nuclei. These dark mauve, purple stained areas are the gray matter of the basal nuclei. So looking at it from the side, these are hidden, the medial and lateral borders of the globus pallidus, but they're all connected, sort of in that lentiform nucleus, that one lump. So why do I bother bringing these up? These are important in controlling the unconscious movements and are related to the actions of the substantia nigra. Remember we talked about that last week. Lesions to the substantia nigra or to the red nuclei and even to the basal nuclei are generally related to uncoordinated movements or inefficient movements, not smooth movements, if you will. Specifically, initiation and termination of movements and linking complex movements together. Now it's worth mentioning what each of these structures is thought to do. We can observe activity in let me just point out, sorry, the putamen. Over here is the outer portion of the lentiform, and then the other two, the globus pallidus, are the internal layers of the lentiform nucleus. That outer region, the putamen, we can see activate before movement happens. It activates before movement happens. So part of the plan to execute a task or to activate certain limbs might pass through this area. You can understand how damage to this area would affect that plan. The caudate nucleus specifically is related to eye movement. We can see that activate prior to and during movements of the eye. And then the globus pallidus, the deep structures, we think are involved in maintaining appropriate muscle tone. So if you have a plan to execute an action, you want to do that appropriately. You don't want to punch the soup can on the shelf as you try to reach it, hit it out of the way. You don't want to throw it against the wall as you take it out. You want to regulate muscle tone to an appropriate level to achieve the end goal. That's largely due to the activity of the globus pallidus. There's also connection here to the emotional system. And this proposed connection is perhaps part of the root of symptoms of OCD or schizophrenia, personality disorders, and some of the spastic activity that happens in those situations. This connection is one that we don't fully understand. We don't understand a lot of the brain. The big question in biology is to understand and decode the brain. Some areas we have a better grasp on than others. This link is one of those ones that we don't have a good grasp on. But we think it relates to personality disorders and the connection between the emotional and the physical aspects of those disorders. So I've mentioned the limbic system a few times, and I'll show it to you here. And you can think of this as the emotional brain. The sensation of feeling largely arises from these structures. And not physical, somatic feelings like pressure, touch, heat, etc. Itches, tickles, things like that. These are fear, passion, happiness, feelings. And there are many aspects, and I only want to draw your attention to two of them. Okay, two main aspects. 2.5, call it 3. First of which is the amygdala, which we think of as the fear center of the brain. The amygdala activates to register the sensation of fear. If the amygdala is damaged, people in which the amygdala is damaged don't show fear or responses to aggression. They don't have that sensation. And the amygdala, I'll draw your attention to at the back, which has a role in creating and storing memories. It's one of the only regions where we observe constant cell division in the brain too, which we think underpins the ability to form new memories. But again, decoding that process is incredibly difficult. Both of these areas together respond to some of the classical neurotransmitters like norepinephrine and dopamine and serotonin, the amygdala and the hippocampus. And it's really overproduction or improper balance or something going on with the response of these areas to those neurotransmitters that we think gives rise to anxiety and depression, mental mood disorders. This is where we might find that physical connection to emotional disorders. And the half, the 2.5, the last one, I just want to draw your attention to the olfactory bulb, which does feed directly into the limbic system, helping to explain why senses of smell bring back such strong memories and sensations and feelings. So a couple key structures to pay attention to. The rest we're not going to explore in detail. This seems like a lot of information. It's really not. That's the bad news. The good news is we're not adding so much new structural information in this section, so that's good. Now we're turning our attention to the application of these structures, the function of these structures, how firing in these areas regulates a certain action, and what these areas even are. Functional organization of the cerebral cortex. Broadly, we can think of it in 3 classes, as there being 3 types of functional areas in the brain. And we already know 2 of them. More good news. We know sensory and we know motor. Sensory are those areas that deal with the processing of information sent from the periphery. Motor executes a task, activates glands, activates muscles, sets a plan in motion. We know those. The association areas are a lot more difficult. They're more complex. And this is where we think things like memory, reasoning, judgment, cognition lie. And in the simplest terms, I liken this to in Inside Out, when the young kid makes the switch from being happy or sad to being melancholy and having both emotions at once. It's the development of these types of areas that I think of in that regard. So association areas. We'll do a couple examples. If I were to hand you a pencil while you had your eyes closed, you could feel the pencil, the surface, the friction, the pointiness of it, and you'd be able to identify the pencil without looking at it. That's the action of an associated area. Or if you're making a lasagna at home and you look in the oven and you see the pan with the lasagna boiling, you got a glass dish in there, you can imagine what it would taste like. You can imagine it's probably hot. You can imagine what the surface of the pan would feel like. It's probably smooth if it's cool. And you can even tap into memories that you form. So the last time you burned yourself, you know that's bad, so you're not going to do it again. You get oven mitts before you take the pan out of the oven. And you think you would learn that lesson once, but I have a good friend that was making a frittata with a cast iron pan as recently as 3 years ago, 2-3 years ago, and he's my age. And he took it out, he started adding some stuff to it, and then he was just caught up and went to put it back in the oven, grabbed and lifted the cast iron pan off of the stove while it was still hot, dropped it immediately, and just his whole hand blistered, covered it in gauze, couldn't use it for 2 weeks afterwards. Doesn't have scars. Incidentally, he used bio oil on them, which seems to work really well. This same guy wasn't wearing glasses and was doing some chainsawing and had some kickback happen where he jumped up and cut his cheek open. Bio oil worked great for that too. Maybe he's just an accident-prone guy. I don't know. But you'd think you'd learn those lessons and then invoke those memories in future. So hopefully he uses more protective gear and doesn't pick up a hot pan. Now there are many different areas that we've been able to categorize and locate and define. And the numbers parallel those areas. These are from Brodmann's original study of the brain in 1909. And we don't care about all the numbers. We care about some of the main ones, but there are many more areas than we're going to look at. The areas that we are going to explore briefly in our class are the primary somatosensory area, the posterior border of the central gyrus, the postcentral gyrus. We're going to look at the primary motor cortex, the precentral gyrus, so that central sulcus dividing the frontal and parietal lobes, pretty important. We're going to look at Broca's speech area, which I think is kind of neat. And then I had a question after class last week about language, and I want to take a look at Wernicke's area for that reason. This is an area that's devoted to learning new language and invoking the use of language you already know, and being able to add inflection and emotion and interpret that in others too. So it's really interesting. So we'll talk about these. Honorable mention to the prefrontal cortex, which is really where we as evolved organisms can point to say this is where reasoning, logic, being critical, debate, all those higher order functions, mood, intuition, things like that are housed, things that make us human. And I noticed in lab the visual cortex in the occipital lobe was one that we referred to a lot as well. So like blows to the back of the head will often make you see stars. The visual cortex, the primary visual cortex, is in the back of the skull. So we're not going to get through all these today, and that's okay, but we're going to start. We're starting with our primary somatosensory area, the postcentral gyrus. And I like these areas, this and the motor cortex. They're intimately related, and you'll find that with a lot of these structures. They often border other areas that are involved in a certain task or processing of information related to a specific task. Primary somatosensory area, if you think about it as a band moving up from the lateral to the medial border, devotes part of that band to each area, each part of the body. And I'll show you that in a second. It's also very close to other relevant areas that are involved in processing information related to the somatosensory afferent signals from the rest of the body. Specifically, itching, tickling, heat, vibration, those physical somatic senses. As an example, the idea of association to other areas. So, if the part of the somatosensory system that dealt with, let's say vision, let's say there's damage to the visual cortex, I won't be able to see part of my visual field, or maybe all of it. There's damage to the visual cortex, I can't register that sense. It's bordered by associative areas that relate vision to other feelings. So let's say the association area is damaged, not the visual cortex. I can see just fine, but I will have no ability to identify a chair or a car or something like that. I won't know what it is. I can see it, but I can't identify or put a name to what that structure or figure is. The homunculus illustrates that division. Homunculus is a Latin word for little man. And this distorted map of the brain is actually not in the edited St. Effects version of the textbook. It's in chapter 16, which is unfortunate. That's why I'm showing it here. I'm also going to put all these updated slides on Moodle like we did last week, so don't worry, you'll have this information when we're done. But this represents the relative parts of the postcentral gyrus, the somatosensory cortex, that are involved in sensations from different parts of the body. Notice all the areas of the body are represented. And the size of the representation is the proportional input from those areas. So lots of sensory information from the hands. Lots of sensory information from the face and the lips. Little sensory information, oddly, from the genitals, which is surprising. None from the knees, really. And all those other structures which aren't terribly important. I'm showing you simply the right hemisphere. So we're looking from the anterior side towards the back of the brain. This is the right hemisphere, a face-on frontal plane. This is paralleled on the left side too. Right? And those senses are from their respective sides of the body. Right hand, left hand, right side of the lips, left side of the lips, right eye, right foot, left foot, corresponding to senses from the same side of the body. So we'll continue with the motor cortex on Thursday, finish this up and get into cranial nerves. Have a wonderful Tuesday. I'll see you later.